Hi everyone, we're Group RMH and this is our final presentation and it's an overview of our gene, the Brady 3G55840 gene. First we have our spectrophotometer results and those indicate just general information about Brachypodium distachyon. Our spectrophotometer results indicate that the sample 1 had a comparatively higher concentration of nucleic acid than sample 2. The 260 to 280 ratio value of around 1.8 indicates that there's a relatively pure DNA concentration, whereas a ratio of around 2 indicates that there's a relatively pure RNA concentration in the sample. The 260 to 230 ratio of around 2.0 to 2.2 indicate relatively pure nucleic acid samples. And the samples without RNase were the ones that were used to conduct the yield for samples 1 and 2. Next, we have some general information about our gene. And first is that it contains 3 exons. This is supported by the fact that the predicted location of the exons, the PASA assembled EST, and RNA sequence log scale all indicate 3 different exons. The location and quantity of the exons are corroborated by these measurements. Contiguous sequences indicate the sequences present for exons 2 and 3, but not for exon 1. A phylogenetic analysis was also conducted on our gene and other organisms, including Ambarella trichopoda, Sinalum lycopersicum, Arabidopsis thaliana, and Oryza sativa are explored through the phylogenetic tree. The closer the organisms are related to each other, the closer they are located to each other on the phylogenetic tree. Knowing this, Brachypodium distachyon is the most closely related to Oryza sativa, but is not closely related to the other organisms. The Oryza sativa gene is most closely related to the gene in question, is the OSNPB 0208221000 gene. The more closely related the organism is to B. distachyon, the closer the organism's gene functionality is to the gene in question. And we created this phylogenetic analysis using the Tree of Life in the ITAL software. Next, we have maize, barley, and rice, which were used to find our gene's functionality and the expression of LSI2. The identification and characterization of maize barley LSI2 like silicon efflux transporters reveals a distinct silicon uptake from that in rice, and the rice and zebra 3 mutation disrupts the citrate distribution, produces transverse dark green variegation in mature leaves, were the research articles that were used to ascertain the functionality of Brady 3G55840. Silicon uptake in barley plants in relation to relative expression of HV-LSI2. This indicated that silicon intake increased with the relative expression of HV-LSI2. Barley and maize have a closer genomic relationship to our gene in question when compared to rice. This information can be used to determine the information about silicon transport in our gene. Silicon intake is important in a plant because it can help the plant adapt to stressful environments and itself can adapt to the environment. The transportation of silicon is more similar to how silicon in maize and barley is transported rather than rice. Maize and barley have silicon in the form of SiOH4, goes through the epidermis to the cortex. After the silicon goes through the cortex, it gets transported to the endodermis into the stele. The stele transports silicon into the body, into the rest of the cell. The Brady 3G5584 gene, our gene, is likely to have influx transporters in the epidermal cortical cells and the hypodermal cells, while efflux transporters are located on the endodermis. The location and types of transporters allow silicon to get transported from the soil to the plant. Maize and barley transports silicon through the epidermis with the help of LSI1, but moves through the endodermis through the LSI2 transporter. On the other hand, rice is transported through the exodermis from the outside to the inside of the exodermis with the help of LSI1, but is transported from the exodermis in the cortex with the help of LSI2, as you can see in our. The chelating functionality of our gene is elaborated further by the figures present on this slide. The figure on the left indicates a dark green plant having a high concentration of citrate in the leaves, especially when looking at the concentration from 90 days and 117 days. When looking at the wild type plants, it was observed that the citrate concentration increased as the plants got older. The figure on the right indicates a healthier plant having more consistent chelation functionality, and the Z3 mutation disrupts the ability for the plant to efficiently chelate throughout the organism. This is seen through the better growth rate present in the wild type plants in the figure on the right when compared to the mutation. This slide and the next slide indicates the gene has the highest expression rate in young seedling stage, and the leaves also support the chelating capabilities of the plant in relation to the gene. The chelation capability for the plant is to form bonds between ions and metal ions. The ability for the plant to efficiently chelate allows plant to transport nutrients throughout the body. Therefore, the increased expression of the gene in the leaves and growing plant would make sense in regards to the gene because the product of photosynthesis and other nutrients would be able to be transported to the rest of the body from the leaves. And the need for the plant to grow, supply, and distribution of nutrients would be increased since the plants would need to grow at its younger state. Here we have our indicated mutation with the highest level of impact. This amino acid changes from a W to a stop codon and will most likely cause the gene to have little or no functionality. And this is due to the fact that it's located on exon 1. Because it's located on exon 1, exons 2 and 3 would not be formed and the mutation would take its effect. Knowing this mutation is important in creating our experiment and exploring the functionality of the gene, which in this case is silicon transport. So here we have our primers for our experiment. The picture on the right is an example of where the primer would be in relation to the entire genome. The double underlined region is the location of the primer, and the portion of the highlighted gene in red is the location of the highly impactful mutation, the amino acid which goes from the W to the stop codon. This experiment is monitoring the uptake of silicon in the roots with the media of silicon in the media without silicon, which is explained in this slide. So we would be growing our plants in media with the absence and the presence of 5 micrometers of salicylic acid. And we would obtain the roots, which is where the silicon intake is the highest, and we would observe the silicon uptake of both plants. In order to do this, we would have to complete a PCR and then have a gel electrophoresis. And based on our findings, we hypothesized that the plants treated with silicon media are expected to have a greater level of gene expression compared to the plants that did not grow in this silicon media. And to conclude, our gene has 3 exons, and research on the organisms allude to silicon transport and chelating functionality of our gene. Our experiment assesses the silicon functionality of the gene is important because this gene plays an important role in plant surviving changes in the environment. which in turn will determine the species' ability to survive in the future. Although much is understood about this gene, further understanding on the functionality and other capabilities of this gene can help scientists better understand how to increase the longevity of this organism, Brachypodium distachyon, and other closely related organisms.